So with that, I will turn you over to our scientific.
All right.
Jon.
All right. Thank you, everyone, for coming. It's a pleasure to be here at UF and the center. Like Don said, I lead the R&D group at BioSkryb, and what I wanted to talk today was our technology to amplify genomes of single cells. And we do so in a way that gives you a new level of confidence in terms of calling mutation that hasn't been seen before. And perhaps even more, I'm excited to tell you about is, as my title suggests, we're going to talk about ResolveOME. And so ResolveOME is a technology to amplify the genomes from single cells using the same technology as I will describe for the DNA isolation. But on top of that, we put a full transcriptome from the same single cell. And as a person who spent many years as a cancer biologist, it's really transformed the way that I do cancer research with my clinical samples. And the idea is that from some of the research that I'll present, I hope it spurs on some insights for your own research. Okay, so by way of agenda, I'll first very briefly talk about why single cell is requisite. I think many of you know this, but I wanted to highlight some key points that I want to reiterate. And then we'll talk about ResolveDNA. So this is our amplification process for amplifying the genomes of single cells. This is also the fundamental technology is called primary template-directed amplification, or PTA. Then I'm going to show you how we've harnessed the power of that DNA amplification and then layered the transcriptome on top of it. And this is called ResolveOME. And I'm going to walk you through two vignettes today to show you the power of this technique. One is with a drug resistance model in leukemia. And then I'll actually show you some data from primary breast cancer cells that I've worked with through a collaboration on Duke. I'll finish the talk looking very briefly at a software suite that we've developed, and our team is working hard to refine to analyze these data. As you know, genome and transcriptome data is very complex. It's intimidating for many folks, and I'll like to highlight how we're trying to ease some of that effort and the power that software suite can impart. And finally, I have just a couple of slides on ResolveDNA microbiome. So we have a technique to amplify DNA, low inputs of DNA in single cells. So we've moved this to bacteria, and there's obviously a big push for microbiome research. So I'm excited to share that with you as well. Okay, so I'm going to start with a single slide on the importance of single cell analysis. And so I'm going to start with the slide on leukemia data in pediatric cancer, looking at mutation rate per megabase. And I really want you to focus on the right is our co-founder and pediatric oncologist, Chuck Gawad, has done some nice work showing that you have basically a 10-fold difference in mutation rate between bulk and what you can see with single cell. So it's very crucial to expose these mutations to understand mechanistically how this is causing cancer. And so this is really a picture of clarity, right? So if we look at bulk sequencing, we're going to miss many of the variants that are contributing to disease, many of the rare variants that are also targetable. And so with this type of amplification and our confidence in calling mutation, we now have a picture of clarity where we can basically understand cell lineage, and we can understand contributions to the disease, cancer and otherwise. And in terms of a single cell paradigm, what I really want to just remind you that if we look at bulk sequencing, we're averaging all of these cells, all of the diverse cells of the population. And in the case of this in silico reconstruction experiment I did, here I'm showing you reads where we've basically spiked in silico one cancer cell in the background of many normal cells. We can't distinguish this green mutation from PCR error from a bulk sample. Now, when we move to single cell, what we have essentially is a digital readout. And this is all I really wanted to remind you about, is that in the case of a heterozygous mutation here, we now have 50% of the reads showing that change, and we have a digital readout, and we're confident in our ability to find these changes. Okay, so what's the core of our technology? So the core of our technology is called primary template-directed amplification. I have a schematic here, but I'm going to simplify this down to two critical points. So this is an isothermal polymerase in green. The cancer genome or genome of your interest is in blue. We're amplifying this as an isothermal process. It's randomly primed with orange primers, but the whole key is this term right here, short. So we're attenuating the size of amplicons. These short amplicons that we create have a lower propensity to be reamplified. So what that does is it allows the orange primers to be freed up. Instead of copying these amplicons, they go back to your cancer genome or genome of interest and result in much more uniform and complete coverage. So another way of saying that is we're preventing copies of copies, okay? And we do that, we make the product short with exonuclease resistant terminators. So it's a very simple process. We're shortening the length of the amplicons. Now, how does this manifest? So in other processes like multiple displacement amplification for amplifying genomes, you have exponential amplification. So say you have a PCR or a induced error at this point, you basically get this haystacking and propagating of an error that may occur. But with PTA or ResolveDNA amplification, you now have what we call quasi-linear amplification, where we have less of this haystacking and we have much more uniform coverage, which in the end, what matters is we now can call mutation in cancer genomes and other genomes. All right, so that's the model. So what do the data look like? So if you retain any picture, I'd like you to retain this picture. This is a chromosome 1, and we're looking at the coverage of multiple displacement amplification across chromosome 1 in 100 kb intervals versus ResolveDNA amplification. Now, while we're not perfect, what you can see is that we have much less peaks and valleys of amplification across the whole chromosome, and we have a much more uniform amplification. This is critical if you're going to call mutation. And so we have much more comprehensive picture that emerges. So what does this do? So in addition to this, we have also genomic coverage. So PTA is this purple plot. Bulk, what we obtain from bulk is in this orange, and I'm showing you as a function of the number of reads, the fraction of the genome we're covered. So PTA and ResolveDNA is much closer to that of bulk, frequently above 95% of the genome that we can recover. And this all matters because we can call mutation. So the sensitivity of mutation calling, which allows us to detect the number of true variants, approaches that of bulk. The precision, again, a way of looking at false positives, is much closer to bulk relative to other methods. So giving you a new level of confidence in calling mutation. What are other critical parameters of the performance? So if we're looking at copy number variation, so if we take a technique like MDA and we compare it to PTA or ResolveDNA, we're much more like the bulk profile. We have much stronger signal to noise. And finally, as a performance metrics, one thing that's crucial, of course, is allelic balance. We want to be able to have amplification product from both alleles. If you don't, you're missing critical factors that might be causing pathogenesis. And so all this is a graph to show you that with PTA and ResolveDNA, this histogram is centered more in the 50% line, showing that we have a much greater balance of allelic representation, all of which is critical for understanding mutation. So how does this work? So I'm going to show you the workflow briefly. So we collect cells. Typically, this is by fax. You can do other methods, whether that be limiting dilution. I'm going to highlight another method at the end where we're actually cell picking based on images. There's a cell lysis step, and then the genome application occurs overnight. And then we have a standard bead cleanup, library prep. Fragmentation is optional at this point. We actually can directly ligate the adapters onto these products. And then we move to sequencing and analysis with BaseJumper, as I'll mention. Another critical piece is not everyone is going to be doing whole genome sequence on every single cell. Right now, the cost is quite high. This is coming down as new sequencers are available. But I wanted to point out that we have a very nice insertion of an exome protocol here. So if you want to do exome enrichment, hybrid capture, you can do exomes on single cells downstream of our amplification. What it looks like in terms of yield, we get about from starting with 6 picograms of DNA from a single cell. We amplify to about 2 micrograms or so from single cells. Here's some DNA controls. And I'd like to point out also that the no template controls are very clean with this process relative to other processes like MDA. And so I walked through the work. Yeah, go ahead. So right now we're barcoding at the library prep stage. So each sample is barcoded at the library prep stage, as opposed to the amplification step. Yeah, so we'll have the cell mapped by library barcode, right? So we're starting with a plate of cells from for example, fax sorted cells. Each well will be mapped by that particular Illumina sequencing index. Does that make sense? Yeah. There are other techniques that we're exploring. That, there's interesting thought of barcoding at the stage upstream of library prep as well that is of interest to us. Yes, exactly, right now. Mm-hmm. Okay, and as I mentioned, we have a BioSkryb library prep kit that can be coupled to this. Okay, and before I move to ResolveOME and start talking about the integration of DNA and RNA, I just wanted to show you, so what are people doing with this? So this is a collaboration that we have with Chris Walsh, a leader in the neuronal field, looking at mutation in Alzheimer's disease relative to normal neurons. And he's used PTA, ResolveDNA technology to essentially refine the rate of Alzheimer's, or the frequency of mutation in Alzheimer's brains versus normal brains. And just some yellow text is highlighting here. He's using nuclei isolated from these brains and then doing the whole genome amplification with PTA. And he did a comparison relative to multiple displacement amplification, saying that, you know, we're mitigating the artifacts that can be seen from MDA. And also these artifacts basically allowed them to reassess the mutation rate that occurs in Alzheimer's disease brains and refine that, drop that down to the rate that they believe is biologically relevant. So as one example of how people are utilizing PTA, and this was published in Nature in 2022. Okay, again, I'm going to move to ResolveOME and RNA, but I wanted to show you, so the data that I've been showing you we have a hand pipetting protocol. As we increase the number of cells, what we've also been able to do is couple automation to this. So this is an HP digital dispenser. It's essentially an inkjet printer that we can use to dispense reagents. And I wanted to impart that by doing this, using different amounts of input DNA, this is a real-time amplification curve of DNA. So this is PTA that has a dye in it, and we're able to track and look at the kinetics of amplification. And you can see for each of these individual amounts, the levels are quite consistent between individual reactions, and it's a nice way of monitoring the kinetics of the reaction. We also can move this to a 384 scale. So just to show you that these are single cells, a favorite cell line of ours that's used that has known mutations across the genome that allow us to benchmark, and very nice reproducibility across 348 cells of this DNA amplification. You also can monitor the quality of the cells with this. So for example, if you have a cell, this is a time axis that amplifies more slowly, you can also discern that. So in that cell that's amplifying more slowly has a poor genome uniformity relative to all of the other cells. It also spikes for chromosome M, saying that this is perhaps an unhealthy cell or some other cell that is not ideal for amplification. We can monitor that. So this slide is to show you that we can move to a higher number of cells, and also with this real-time monitoring technique, it's useful for understanding your system. Yes, let's pull it up. The rRNA amplification. Yes, so we don't have any, the question is, how do we avoid rRNA amplification? So we know that, that in case of mitochondrial DNA, circular DNA amplification is efficient, and so we use that as a gauge to monitor the cell health. We don't do any type of RNA depletion right now, but have not been discouraged in terms of our transcriptomic data in any way by the levels of RNA that we are there. So we're not depleting it in any way, but I'll provide data to show you that we're confident in our DNA signal and our RNA signal despite that background. All right, let's move to the next slide. So am I frozen? Okay. Nice. There we go. There's a delay. Okay, so I've shown you ResolveDNA amplification, and so with that, yeah, I have kind of what dictates cell fate, and there are several things that I have listed here that you can expose with primary template-directed amplification or ResolveDNA. Single nucleotide changes, copy number variation, gene translocation, tumor mutation burdens. Some of these I'll highlight in my biological examples. But now, what if we were able to harness the power of gene expression? So that's all DNA level. I think you can imagine the power if we couple transcriptome to the single cell data, what that can tell you. And as I go forward, I'm going to give you examples of from my patient samples, what we can see in isolation by these DNA and RNA alone, and then what can only uniquely be seen when we harness these together. So that's also shown in this Venn diagram, where I list some things that we can see at the DNA level alone in isolation, some things we can see at the RNA level alone. But I'm going to highlight some things today, like how do we link ploidy and gene expression together from the same single cell? How does that differ from single cells? How do we look at cell states coupled to genotype from a heterozygous or from a heterogeneous tumor? these are things that I'm calling uniquely ResolveOME. And again, with this technology, we're able to look at these things that haven't been able to be seen before. As examples of ResolveOME, very briefly, neoantigen discovery. Look across the genome for new neoantigens. Are they expressed? You can now do that from the same single cell. Gene editing. You can monitor where your edits are. Or you have off target. How is expression altered? How does this vary from single cells? EMT, cells that have become invasive. What are the changes that occur? What RNA changes that occur coupled to what DNA changes? You can't fully understand mechanism without understanding these from the same single cell. Another example, isoforms, P53, couple mutations with isoform expression. And so these are just very quickly some ideas of how you might be able to utilize ResolveOME. Okay, so how does this workflow differ from the ResolveDNA workflow? So what we do is we essentially have a reverse transcription step. Is DT, it's poly A enriched. And we have a way to purify this RNA from the DNA sample. We do reverse transcription first, as outlined in this diagram. And then follow that, we essentially run that ResolveDNA reaction on top of it. So in the same pot, we have the DNA full genome amplification reaction that occurs on top of that cDNA synthesis. And following that, we then able to fork this protocol. So we purify the amplified cDNA or the RNA using an affinity tag. And then we separate the genome fraction and we construct two different libraries. We sequence these, often we sequence them together. And then you have two sets of interpretation: transcriptome from your cell of interest, and you have genome from the cell of interest. Yeah. Right now, so FFP poses some challenges in terms of the amplification. We very much are working on this and want this to be successful because we know that everyone has this, and including our own samples, we want to see that. But right now we have some challenges with the PTA amplification, but also hints of success of making this more accommodating to that. But the data I'm showing you today is from non-fixed samples. Yeah. So there's, we're comparisons and kind of techniques on its own to just to move. DNA also is quite damaged with FFP, right? So challenges abound for everyone in that space. But again, encouraged by some success with some chemistry modifications to try to get through some of those regions. And we're looking right now to see, well, how well are we doing? What's represented? How, are we able to call mutations in this background of damage from FFPE? So it's not a unique challenge to us, but something we're very cognizant about and want to drive forward. Okay, so the two vignettes I'm going to give right now. The first one is, again, I'm trying to show you what we can do now that's separate from isolation, that's only through this unified information. The two vignettes are, I have a leukemia model using cell line called MOM13. It's a model of drug resistance. And then I'm going to show you primary breast cancer work through a collaboration that we're doing and continue to do with Duke. Much of what I'm going to show you today is highlighted in a preprint that we've submitted recently, describing the ResolveOME technique and biology that we've been able to glean. And as I go through these, I will first, again, show you isolation and show you how we're harnessing these together. So I need one quick intro to this model. I'll keep it simple, but this is a leukemia cell line. It has a mutation from which the cell line from which this patient was derived has a FLT3 mutation. So it's an RTK that drives cell growth through ERK and PI3 kinase, right? The mutation is an internal tandem duplication, which is an in-frame duplication that drives signaling. So this results clinically in poor prognosis. Because of this mutation, we can add a FLT3 inhibitor. These cells are exquisitely sensitive to this, of course, but ultimately, like everything else, it becomes invariably resistant. This is, I've created two models. If we look at time, I've created one model where I've dose escalated this drug to inhibit FLT3, and one where I've just basically slammed it and kept it at a little bit above the IC50 for the whole time, and created resistant lines. So what can we see with ResolveOME? So I'm going to show you single cell data from ResolveOME. First, looking at some DNA contributions to resistance. Secondly, looking at some RNA contributions to resistance. And thirdly, how we're looking at the unique insights together. So at the DNA level, first, before we talk about the unified insights, these cells, resistant cells, have vastly different copy number profiles than the parental cells. Each one of these lines is a single cell, and I've colored the single cells by parental or resistant. I'm highlighting 2 chromosomes because they're pretty interesting. Chromosome 5 in the parental cells, they're vastly aneuploid, but we see in resistant cells, we now have lost much of this gain of copy number that occurs in the parental cells. Likewise, with chromosome 19, we have gains of chromosome 19 in the resistant cells, but not in the parental cells. So the cells are responding by copy number changes. But what about with our technology? We'd not only see copy number, but we have the genome application in such a way that we can call across the entire genome, unlike methods before. So if we do that, what do we see? So one of the things that I identified in these resistant cells, we have a single nucleotide change that came out of an analysis in the molecule itself. It's a secondary mutation. So in addition to CNV, we have SNV contributions to resistance. And if we look at single cells by this technique, in the resistant cells, it has that secondary mutation, whereas in both resistant and parental, it has the primary, of course. So it's one way that the cells are driving resistance. Coincidentally, or for more information, this is most likely affecting drug binding based on where it is in the crystal structure. I won't go into too many more details other than to say that between these models of resistance, there's drastic differences in the types of changes that we see. So the copy number changes that we're seeing are quite different between the models, which is not surprising of how we dose these. And again, we also see that mutation, that secondary mutation, but now it's not in every single cell, it's in a fraction of single cells. And interestingly, the cells that have these aberrant copy numbers that differ between the models lack that secondary mutation. So it's like this, the cells have distinct modes of resistance, and depending on the nature of that variation. So that's a DNA level change with this model. And I wanted to show you this briefly before I move to RNA, because you look at this heat map, you think this is a transcriptome. This is not, this is DNA. So the genome matters and is not static. So basically in this model, we have 6,000 or so differentially prevalent single nucleotide changes in the parental single cells, which are each column parental single cells versus resistant. I'm showing you the genotypes across SNVs, across the genome, and how they differ between parental and resistant. So you have a class where you have a homozygous wild type change that in the resistant cells across all of these different lines of genotypes now is heterozygous. So the genome is not static, and we can pull that out with this technology. Okay, so let's move to RNA. So RNA with ResolveOME. So these are single cells again at the top, parental versus resistant, and on the rows we have different transcripts. I'm just going to highlight 2 brief things before I move to the unified insights of ResolveOME. But we have a molecule Gas6 that's upregulated uniquely in the single cells of resistant cells versus parental. This is a ligand for another RTK called AXL. Known method of transcriptional bypass. So there's RNA modes of resistance as well. So in addition to the mutation that I described to you that occurred at the DNA level, we now have a ligand that's upregulated to drive growth through another RTK because FLT3 is inhibited by this drug. Another mode of RNA resistance, we have enhancer factors that are coming out in the resistant cells. So there are epigenetic changes that are occurring as well. So on top of these DNA and RNA changes, what can we do with ResolveOME now? So one more figure before I show you the unified, but I wanted to show you about the nature of the RNA of ResolveOME. So it's not 3' end counting like 10x genomics, where you're only going to get transcript counts. We have beautiful 5' to 3' gene body representation across different models. Here's 2 housekeeping genes, actin and GAPDH, showing you that 5' to 3' representation is exquisite for this transcriptome data. So it's full transcript data. And with full transcript data, you can get insights like this. So we can look at isoforms. So with just end counting, you cannot do this. So one with ResolveOME now, this is an example in my resistance model. I'm showing you 2 different genes or transcripts, PP and HA, 2 isoforms for each of these genes. And the blue versus tan is parental versus resistant. So what you can see is these resistant cells, each of these single cells are the dots and reflected by this box plot. But the resistant cells are utilizing this 5' exon that's not in that the parental cells by and large do not. This other gene, the same thing occurs. There is a parental cell utilization of a longer isoform for this gene that is different from the resistant. So wanted to highlight that full transcript data allows you to pull out that richness of information that cannot be associated with N counting. So with that background in this model, and before I show you the patient data, well, how do we unify the data? So we're talking about ResolveOME. So what insights are missed by looking at DNA, RNA alone? So this is a graph of ploidy, essentially. So there's different classes of let me back up. I'm sorry about that. 2N normal copy number here for 2N, and moves to greater than 6 because these are aneuploid cells, or reduces or loss of copy number to 1N. And on the top, or on the rows, I'm showing you particular loci or genes or transcripts. And then I'm showing you whether the expression is gained in red or lost in blue. So now we can look at DNA level information and how does that coincide with RNA level in terms of transcription. So you can see interesting things. So you have expected things, right? So in the single cells where we have ploidy that's up, right, 6N or whatever, expression is up. That makes sense, right? But we have cases also, this also is directional where ploidy is down and the expression is down. However, interesting classes emerge that you cannot see in isolation. So in these cases, this expression is down, but the ploidy is up for this particular locus. Likewise, we have cases where ploidy is down and the expression is up. So a way of linking the DNA information at the single cell level that cannot be seen in isolation. So, when we have discordance like this, could this mean epigenetic mechanisms? Could it steer you in a different direction than you would have done otherwise? All right, so what about further ResolveOME insights? So in the cases of you have a favorite oncogene that you say is upregulated, how is this transcriptional upregulation occurring? Is it epigenetic, like it maybe alluded to before? Or is it a DNA variant? Is it copy number? And so now we're in a position to answer this really powerfully, and I'm really excited about this next type of data. So a little bit of complicated of plot, but I think it's very insightful, so I want to walk you through it. So what we're essentially doing is, if I can hide the zoom, I'm sorry, a little bit. Or maybe I could minimize. We're looking for regulatory variants, right? So with ResolveOME, because we have the whole genome amplification, technologies before would give you partial genome coverage or give you really poor allelic balance. You can't be confident that you're calling all the variants. So what we can do is we can say, we're now in a position to say, are there unique DNA changes that affect transcription? So what I'm showing you in this plot is a bubble plot between parental and resistant single cells. The color of the circle refers to expression level, so at the RNA level of this particular gene that's on the rows. The size of the circle refers to a genotype. So again, homozygous wild type, homozygous mutant, or heterozygous at the locus of this particular gene. And then we've looked at loci that are promoter loci. So what are DNA variants that are close to the gene that might be influencing expression via promoter mutation? I've highlighted some that are here in the gene body. We're moving to enhancers, of course, looking at loci across the genome that are causing expression differences. So what do we see? So with ResolveOME, we can start looking at these data. If I have a delay, sorry for my animation. All right. It's not advancing. 1 second. All right, hopefully I'm not frozen. All right, wonder if I can. Oh, there we go. Delay for some reason, I apologize for that. Okay, so MYC, right? Screaming oncogene of importance. Look at MYC. I have the animation off. Okay, we'll look at MYC first. So MYC in resistant cells, great difference in expression. Parental, lower difference in expression. This coincides with the genotype. This particular genotypic change happens to be a single nucleotide change that differs between parental and resistant in the intron of this gene. This enhancer factor up here, or actually it's a splicing factor, a poly A binding protein. Same thing, we have an expression difference between parental and resistant single cells. that emerges between red and blue, there's a variant that's within 5 kb upstream of these cells that differs between parental and resistant. Is this a promoter change that is causing that RNA expression difference? So I don't know for sure if these are functional, but that's not the point. The point is this method exposes those changes at the DNA level that influence expression and now have the ability to screen genome-wide and look for regulatory variants from single cells and how that differs. Okay, so let me move in the closing part of my talk away from the cell line model, and I want to move to primary samples using ResolveOME. So this is a collaboration with Shelley Wong, Chief of Breast Surgery at Duke, and Jeff Marks in pathology. This particular patient I'm showing you is a ductal carcinoma in situ patient with elements of invasive cancer. So the question we're asking biologically is, okay, as this cancer goes from a relatively benign state to invasive cancer, what are the genomic and transcriptomic events that cause this transition? You can only do this with in my mind, at the single cell level, and you only can do it if you have both RNA and DNA information. So how we've done the study is we've enriched for EpCAM high or epithelial cells from these singulated mastectomy samples, put them into wells, and we've run ResolveOME on them. And we're now I'm going to present you some insights on ResolveOME using again delay. I'm sorry. Let's try spacebar. All right. It's not advancing either way. If I have to back out, maybe. These are fresh frozen mastectomy samples from a biopsy. Oh yeah, let me pull it up. Let me pull up the. Let's see if I can share the screen for Zoom. No. Sorry, guys. Okay, yeah. So these are singulated cells that I obtained from mastectomy specimens. So in that unique case, so they're not FFPE in this case. All right. So again, looking at these cells, one of the questions I've always had is I find a mutation. How do you know that's really a cancer cell? What are the other cell types that are there? And so now with ResolveOME, we have a transcriptome. We can now answer that question from single cells. All right. So some quick insights. So really amazing copy number loss in these cells in tumor versus normal biopsies. Losses that make sense in terms of tumor suppressors. So deletions on chromosome 13 with BRCA and RB, deletion on 16 and 17 P53. And many of these have been seen before in DCIS. So we're exposing these from single cells. And if you notice, they differ between single cells. So the cell has 2 deletions, the cell has 3, this has 3 different. And so we've been cataloging these as we move to more single cells. It's really amazing to see the genotypic diversity, even with this low number of cells. That is highlighted here. So with this DNA level, before I move to the RNA joint insights, we also find mutations, so PI3 kinase mutations. Interestingly, we see that the copy number loss coincides with one mutation, but not the other. And this could only be seen from single cell data. Again, each row is a single cell. You can very nicely see that. So even deep sequencing 30 cells from this mastectomy sample, we have this ball of grapes diversity of genotypes that occur within this patient's tumor that we now can see and expose. We have additional patients. We're moving to 11 for this study. This slide just shows you that there's different mutations between patients, and the copy number profiles look very different between patient 1, patient 2, and patient 3. One element before I move to the joint data is that we've also been finding what look like oncogenic mutations in normal biopsies. So this is a ipsilateral breast control sampling. So the biopsy needle was in the same breast next to the tumor. Found a very prototypical PI3 kinase mutation change in the normal tissue. Could this be a crossover border event between the normal biopsy and the tumor, correct? But also, I think this is starting to show us that we can capture pre-malignant events in these cells. Likewise, in this case of a contralateral breast sampling, so the other breast, we still see what look like very characteristic DCIS copy number changes that occur in single cells in the normal breast tissue. So really excited about the possibility that technologies like these can expose these what are likely pre-malignant events. All right, so I'm going to finish the breast cancer story with this figure. I really love this figure. This is now taking ResolveOME, so I've shown you some DNA insights, but let's add RNA on top of this from these single cells. With my future samples, I'm running exclusively ResolveOME. I would only do DNA before. It's hard for me to go back now. I've become addicted to the data, and you get these insights and you don't want to go back to only looking at them in isolation. So this is an RNA heat map, transcripts on, and it's only a portion of this heat map. I spliced it off. But genes that are significantly different between the epicam high population that I showed you we sorted on, which we believe are tumor cells, versus an epicam low population. So what can you see from this multi-omic heat map that's caused by that we can generate by ResolveOME? So we have multiple tiers of information. So the heat map itself is RNA. So at the RNA level, you can see things like, well, look, FOXA1 is upregulated in the cells that we believe are enriched for tumor cells, a prototypical breast cancer transcription factor, correct? But in the epicam low cells, it's very low because we have a diversity of cell types in the epicam low that came down from this mastectomy sample that I can only see now with RNA information. So for example, we go to the next level. Before I get to that, let me show you C and V. So we can layer the copy number information on top. We can see on top of the copy number, we have the PI3 kinase status. So I showed you that mutation. Well, look, that mutation in these single cells, each column is a single cell, is almost exclusively in the epicam high population. And in the epicam low population, it's almost exclusively no mutations. So I think that says that we did an okay job fact sorting in this case, but it shows you also the diversity of cell types that are there now that we have transcriptional information from the same single cell. So we have endothelial cell that appears. We have a fibroblast. We have a monocyte, immune cells. So all of that can be seen now from this type of data. One other thing that I'll stop the breast cancer story after this, but then I also want to think about cell state. So not only cell type, but what about cell state? So look at this one interesting cell, single cell that is here. It's in the epicam low population. We assumed it was not a tumor cell, but it shows up as an epithelial cell, and it has a mutation that suggests that it might be cancerous. However, it clusters expression-wise in this cluster in the epicam low population that is more fibroblast or stem cell-like, and that can be seen from the cell identity matrix, where look, now we have expression profiles that, although it has those oncogenic mutations, now looks more like a tissue stem cell or a fibroblast. And so I'm very excited, and I think this is going to be a theme going forward with people doing this type of work, is that you can now make, start to make cell site insights into this tumor paradigm. All right, and for example, is that cell entering EMT? And I really do believe that we have insight into cell state here. Okay, so ResolveOME, we have an early access program for this right now. If it could be useful to any of your research, please talk to Don and Katie. I'm completely excited that this is in other people's hands. I'm very much looking forward to data other than my own, but I've been pleased by my data and how it's been able to transform my own data. As I look to close, let me give you a couple more things. So all of this has been fact sorting so far. So how do we go to the next step? Spatial is huge, of course, right? We want to know, can we be guided by an image? So can we take a tissue section? Can we pick tumor cell versus stroma? Or if you're interested in the immune genotype, can you pick your immune cells? So we have a partnership with ALS out of Germany. They make this cell selector. It's a really cool instrument. There's a microscope, and it has a picking mechanism. So you can take your image. It will pick that individual cell that you marked, not unlike laser capture microscopy, and you can do ResolveOME on it. So you can, here I'm just showing you that this is a capillary that's picking those single cells. We can put them in a well and run our chemistry. This is an experiment to show you empty wells versus picking cells to show you that we have lack of carryover there. And this is ResolveOME data. So we have an RNA fraction data and we have DNA fraction data from this picking. So we can move to spatial at a level that can't be done with facts. Yep. Quite honestly, sometimes we'll get two together. They'll be a little sticky, depending. You can image that. There's actually before and after images that you can look at that. And in those cases where you do have, I don't know, maybe epithelial stickiness between one or the other. So this is actually not a there's lasers for just the fluorescence imaging, but it's actually physically sucking up that cell. So we do some treatment to kind of loosen it a little bit, and then we can monitor and watch it. So we're always guided by the image. But you're right. There are cases where every time is not a perfect single cell. We'll have a straggler coming with it. Yep. Okay, and I have a few slides on BaseJumper. So the data are complex. It's intimidating. I know a lot of projects have stalled because potentially for the money of whole genome sequencing, but also the analysis, people can be intimidated. So all the slide is saying the data are complex, but we're also working to enable people to do some of this that don't have their own bioinformatics solution. It's called BaseJumper. It's a cloud-based system. Vic Wigman, our computational director, has done a great job putting this together. I'm just give you very brief highlights of what it can do. All of your QC reports from each individual single cell. You have IGV viewing. You can look at each individual cell. Copy number plots come out automatically, like I've been showing you. You can zoom in on a particular region. Oh, this is interesting. What about this dot? Is this a gene that I care about? It'll pull out all the genes that are in that amplified region for you and bring them to your attention. Variant filtering. We're looking at whole genome data, extremely complex. The VCF files are huge. This is a way to condense that, bring that down. Things like we can look at nomad databases. Is this a change that's seen in the population? Predictions on protein function. Is this disrupting protein? And other things, yeah, by position, and just allowing you to simplify a VCF file to get at the variants that you care about mostly. Because it's ResolveOME, we have RNA on here, so everything that you would typically do, you would have, you can look at QC metrics, you look at heat maps. It can do PCA plots for you, and volcano plots for differential expression. All of that's built in. Many of these common things that you want to survey your samples for initially and see how it looks. And so I'm using this tool internally as well. It's for me, it's cut out a lot of middle steps. I'm not spending my time at the command line, and I can do this relatively quickly. And the last two slides I'll show you before I close. So I've shown you cancer genomics, right? I mentioned neurology with that collaboration. There's a lot of other applications that you could quite easily see, I believe. Pre-implantation genetic testing is a big push for us. We have a lot of collaborations there. Other things, so microbiome, right? So we start with a small amount of DNA. The attributes of PTA that we use to amplify the DNA well from a mammalian genome are still useful in the context of bacteria. So I won't belabor what you can do with full bacterial sequencing, but some of the things that I'm involved in now, we're moving to clinical samples for microbiome using our amplification technology, gut and vaginal, what we're focusing on. And I'll give you an example of a novel environmental species identification. Basically the same workflow, but we have a little bit of stuff upstream to lyse the bacteria well. This is data that we generated internally. These are just fact sorted. My best attempt at single cells of bacterial fact sorted E. coli and B. subtilis have grown in the lab. These are plots that show you contig assembly and how complete we're getting to the genome. If we're getting to the estimated complete genome, you see that's that dotted line. We can do this quite well for B. subtilis for E. coli. I can take a mixed sample. We can deconvolve the mixed sample. And of course, we're trying to do this on more complex samples, non-lab grown samples, and in real environmental or clinical samples. So one collaboration that we've started with, Nate Sierra at Cornell, he's interested in the biology of these salt marshes. And so hidden a little bit by this zoom, but he considers the salt marsh very much an organ. So these species interact and communicate, and he's interested in dissecting that at this whole genome level of single bacteria. So some data initially where he simply did limiting dilution of these salt marsh samples, ran ResolveDNA amplification, and from his first experiment believes that he's identified new genera from the salt marsh. So very excited to see what collaborators and customers are going to be generating the type of data from the microbiome as well. All right, space bar. Apologize for these freezes, and this is the last one, hopefully. Let's see if I can. I think again, it's uniformity of amplification and completeness of amplification, right? So there are other techniques to amplify genomes. So MDA, for example, so they've been doing MDA and they've been quite pleased to see the difference. I mean, again, it's completeness and uniformity that allows you to call these variants, and in the case of bacteria, complete the genomes, right? So we've been doing some pretty head-to-head comparisons between the two methods. All right, let me stop this and get to my closing slide if I can escape out of it. All right, let's reshare. Okay, so to finish, I'd like to bring this figure up again. Think about uniformity and peaks and valleys of coverage. And if you care about DNA variants in your system, you need a situation that is like this, or that is like the blue, right? ResolveOME. So it's changed the way I do my cancer research. I still like to think that's at least a part-time job for me. And our collaborators are doing this right now, and people are our customers. I've shown you a leukemia model of resistance, talking about a couple things. Genomic penetrance are the changes we see expressed. Regulatory variants, we have a new way to discover these. In the breast cancer example, I've shown you examples of how we can now pull out cell type and cell state in the context of mutation and CNV data. And lastly, a hint at BaseJumper and its powers to help with analysis for those that need it. And I finished with some microbiome work. So thank you so much, guys, for your time. And really, hope it put in your mind at least about if DNA and RNA information from single cells is useful to you, how it might be, how it can help you in your system. And happy to take any questions. And again, thank you for your time today. Yes, I was about to take a peek. Okay. Yes. So the question is, maybe I'll type it in case it's not on the Zoom. How do we separate cDNA and gDNA libraries? So we use an affinity tag for that. So it's a biotin system. So we can use biotin streptavidin to pull those out. The RNA is again, it's poly A based, and it's template switching based reverse transcription. Not unlike SmartSeq, but that's how we do the purification. I'll type it in case it's there. Right. So right now we sell 96 well kits. Of course, a thrust for us is moving to higher throughput. You got a glimpse of that from the 384 automation that we're doing. We're also starting down the road of trying to get a 1536 type scale. What I also want to leave you with is that all of that breast cancer insight, and I'm seeing some pretty amazing things at 30 cells, right? So is that going to catch every variant? No, but we believe that kind of this sweet spot of 96-384 is going to be very useful for many cancers. But we're working on the automation and coupling that so that we can go to that level for customers that need that level. And it's going to vary by system too, right? I mean, I guess it depends on what you're trying to see and the frequency and how rare your mutation is. But we feel kind of 96-384 scale is going to open up a lot of insights. I'm using a Sony SH800, and I'm not claiming to be a single cell bacterial expert, but we have some computational proxies for estimating cell number in there as well, and using kind of contaminant DNA as a proxy for standardizing that. And there are cases where we believe we have more than cell in a well, but I think for our microbiome community, and you can tell me as well, that's probably not going to be a problem. I think for the mammalian community, they care about that single cell more crucially. But what I'm doing with the clinical samples and the marsh samples, that even if we have 5 to 10 cells in there of different species, genera, or whatever, we have the computational ability with our team to extract those. The genomes are complete, and we feel that that information also is going to be quite valuable to the microbiome community, even if it's not exactly a single cell. But yeah, it is challenging technically to do that. I've had some success, but I'm by no means perfect at it. Yeah, I think that's exactly the case with the microbiome. And again, I think the sweet spot for that is limiting dilutions or other techniques where you get 5 to 10 cells in a well from a clinical sample or environmental sample, and deconvolve those and start to make insights across the plate. Yeah. Yeah, sure. Yes, yes. Illumina adapters, right? Yes, so it's at the library level right now. I mean, there you can imagine there's could be great utility in barcoding at the amplification stage in terms of pooling and those are things that we're interested in. But right now we're barcoding per well with a Illumina index. Yeah. The structure, well, so right now, again, they're just Illumina adapters, right? So that's how we're barcoding. We don't have barcoding for at the amplification stage. I've showed you that the primer we're using is a randomer right now. We don't have barcodes on there. We don't have a UMI, no. Right now it's well based and with the and that's something we currently don't have that on the transcriptome, but that could be not a hard addition. Yeah, so single cell per well, right? And then one library is generated from that single cell from that well, and one Illumina index associated with that right now, right? So it's still plate based. Again, exploring other ideas to improve the throughput and workflow, but right now that's how we have it structured. Yes. Mm-hmm. Yep. So it's run the PTA, run the ResolveOME, and then have that output product created a library in a different well with a unique barcode. Yes. To track the sample. And then of course with like index sorting and FACS, you can then track back to, oh, do you have a cell that has an interesting genotype or transcriptome? You can go back to the FACS plot and see, oh, is this lie in a different spot of your FACS machine gate 2, and maybe there's some genotype phenotype correlation you can do. Yeah. Any other questions? Don and Katie in their back, if you have further questions, or if you can come up talk to me and also contact us by email if you have any interest. But again, thank you so much for your time, and it's a pleasure to be on campus.